Assalamu alaikum. This is Dr. Mamoon Ahram. I'll be teaching you, I'll go over this course with you. As you know, this course is divided into three parts. The first part is molecular biology. Then I'll go on with you into cell biology, a few lectures on cell biology, and we will cover genetics at the end. Okay, so this is the molecular biology part. We'll talk about nucleic acid structure. I'll define first what the molecular or central dogma of molecular biology is. Then I'll go over gel electrophoresis, southern blotting, a few techniques, followed by description of what the human genome is all about, DNA replication, mutations, DNA repair. Then we will go into the process of transcription, regulation of transcription in both prokaryotes and eukaryotes, and then we will talk about a few techniques regarding regarding gene expression, and then we'll go over translation and its regulation. So I'm gonna depend on Cooper's the cell A molecular approach, which you can get, but the main resource is the lectures themselves. So for this lecture, you can refer to these few pages in Cooper's. Now, nucleic acids are divided into two types. We have deoxyribonucleic acid, also known as DNA, and we have ribonucleic acid, which is known as RNA. Now, what the central dogma of molecular biology is that it tells us that there are different processes, starting with DNA. DNA can replicate itself, so a copy of DNA is made from DNA. Human DNA is double-stranded, meaning that there are two strands of DNA. From DNA, it can be used as a template, okay? So focus on this word, a template, in order to make an RNA molecule, which is single-stranded. This is a process known as transcription. Then we have translation, whereby RNA is used to make protein using ribosomes as well as other molecules. So this is the basic central dogma of molecular biology. Now the thing is, after this was initiated, or after scientists came up with this, they noticed that actually the process is. So in reality, that's how it looks like. Yes, you do have DNA replication. A copy of DNA is made from DNA. You do have transcription, whereby one strand of DNA is used to synthesize a single-stranded RNA molecule. And you do also have translation. Protein is synthesized from the RNA molecule. Now the thing is, in especially viruses, RNA can be used as a template to produce DNA. This is a process known as reverse transcription, and the enzyme is known as reverse transcriptase. In addition, also in viruses, RNA can be used to make an RNA molecule. So a template, you have an RNA template that is used to make an RNA molecule as well, a copy of it, a complementary copy of it, in a process known as RNA replication. This actually is the process that takes place, that coronavirus uses. So the coronavirus genome is, and the genetic material is RNA, and it makes RNA copies of itself, of its RNA molecule. So nucleic acids in general, they are linear. They are polymers of nucleotides, and these nucleotides are the monomers. So nucleic acids are polymers because they are made of repetitive small molecules known as monomers or nucleotides. And these nucleotides are linked to each other via a phosphodiester bond. Now the thing is, DNA is coiled, that is, it's folded on itself. It's a very complex process, so that it fits into the nucleus. Okay, and usually it is associated with proteins, forming a large genetic material known as chromosomes. And we'll talk about this. So let's focus now on the monomers, on the nucleotides. So any nucleotide is made of three components. One, you have a sugar. Two, attached to the sugar, you have a nitrogenous base, and this base is linked via what is known as a glycosidic bond to carbon number 1 of the sugar. And then you have, on the other side, you have a phosphate group attached to carbon number 5. Okay, so overall, this molecule is acidic because of the presence of a phosphate group, and this phosphate group is negatively charged. So the whole molecule of nucleic acids is actually an acidic molecule, highly acidic molecule. Okay, now there are two types of sugar molecules that make up nucleotides. The first is the first is known as a ribose, okay, whereby it's a pentose, that is, it's a 5-carbon sugar known as a ribose, and that's how it looks like. Now, and the other one is known as a deoxyribose. The reason is that this hydroxyl group of carbon number 2 is actually a hydrogen, not a hydroxyl group. Okay, so that's a deoxyribose molecule, and this is one difference between DNA and RNA. That is, DNA has carbon number 2 as a hydrogen versus RNA having the hydroxyl group attached to carbon number 2. Okay, now I said that nucleic acids are largely acidic because of the many negative charges introduced by the phosphate group. So these nucleic acids are overall, they are stable, but because of the many negative charges, they can have large degree of repulsion. And as a result, these nucleic acids can be stabilized by the addition of positively charged ions like sodium ions and magnesium ions. So these ions mask the negative charges, reducing the repulsion, and as a result, stabilizing nucleic acids. Now, in eukaryotic cells, but not in prokaryotic cells, DNA is complexed with proteins known as histones, and these histones are positively charged proteins. So this is how they can interact with DNA. Now, notice something here. Notice this sign right here. Okay, this is 1', 2', 3', 4', and 5'. So why is it that we have prime added to the number of carbons in the ribose, in the sugar molecule? The reason is that there are also carbons that make up the nitrogenous bases, and they are given numbers of 1, 2, 3, 4, etc. So basically the idea is that to differentiate between carbons of nitrogenous bases versus carbons of the sugar molecule. Now pay attention to this one right here and this one right there. This is the 3' carbon and this is the 5' carbon. So pay attention to that because we're going to get back to it. So these are the nitrogenous bases. In fact, there are two types. There is a type known as a pyrimidine, and there's another type known as a purine. And the sugar is attached to nitrogen number 1 of pyrimidines and nitrogen number 9 of purines. Notice the difference in structure. Pyrimidines are single ring molecules, purines are double ring molecules. Okay? And one way by which I can differentiate between purines and pyrimidines in terms of structure is that purines, the little word purine, is a large molecule, double ring structure, versus pyrimidines, large word for the small structure, that is the single ring. There are two types of purines that make up DNA. and RNA, and these are adenine and guanine. Now notice the difference between the two. Adenines have primarily have a nitrogen and amino group right here, and guanine has a ketone group right there. Okay. The other difference is that guanine contains an amino group also attached to carbon number 2, but that does not exist in adenine. So you don't have to know the exact structure of guanines and adenines, okay? Just know that adenines and guanines are purines. Purines are double ring molecules. Now, both adenines and guanines also exist in DNA and RNA. In terms of pyrimidines, you have cytosine. Cytosine has an amino group right here, as well as a ketone group right there. Okay. Now, cytosine also exists in both DNA and RNA. But here's another difference between DNA and RNA, in that in DNA, thymine exists in DNA versus uracil, which exists in RNA only. So it's not normal to have uracil in DNA. Okay, the difference between these different pyrimidines is that cytosine has an amino group right here versus thymine, which has a ketone and a methyl group. Uracil is very similar to thymine, but it does not have this methyl group right there. Okay, so this is a large difference between the two. Okay, now, so these are the bases. Okay, so there are differences between DNA and RNA, and that is DNA is usually double-stranded, RNA is single-stranded. DNA is a deoxyribonucleic acid, deoxy because it has a deoxy sugar at carbon number 2, 2 prime, it lacks a hydroxyl group, versus RNA, ribonucleic acid, it has a ribose group. Okay. Another difference is that thymine exists in DNA versus uracil, which exists in RNA. Okay, so these are the sugars, the bases. And now, what is the difference between, what's a nucleotide exactly? So a nucleotide is a molecule that is composed of a sugar, whether ribose or deoxyribose, a nitrogenous base, it can be a purine or a pyrimidine, as well as a phosphate. And it can be a single phosphate molecule, 2 or 3. All these three things, these molecules, are known as nucleotides. So in order to differentiate between the three molecules having 1 phosphate, 2 or 3, we use the word nucleoside, the term nucleoside. So what is a nucleoside? It's a molecule that is composed of a sugar and a base only, without a phosphate. Now, if we add a phosphate, it's a nucleotide. And to be more specific, we say this is a nucleoside monophosphate. So if I say a nucleoside, I know that it has a sugar and a base. If I add a phosphate to it, now I know what the number of phosphate molecules attached to this nucleoside is. Okay, so it's 1, I say monophosphate. If it's 2, then diphosphate. If it's 3, then triphosphate. Now, a very famous example of a nucleoside triphosphate is ATP, which I know, I'm sure you know of. So this is how we name nucleotides and nucleosides. I can say deoxyadenylate, and this means that the sugar, 1-deoxyadenylate, it's a deoxyribose right here. Adenylate having an adenine group right here. If I say deoxyadenylate, it means that it is also a nucleotide because it has a phosphate, just 1 phosphate. Or I can say deoxyadenosine. So adenosine means that it is a nucleoside, monophosphate. Or I say it's AMP or DAMP for deoxy. The nucleoside is deoxyadenosine only. Or I can say deoxyguanylate, I'm sorry, if it has a guanine, or deoxythymidylate, or deoxycitidylate. Now, if I'm talking about the nucleotides that make up RNA, I can say adenylate, because now it's not really deoxygenated like in here. The ribose sugar has a hydroxyl group at carbon number 2 of the sugar. So adenylate, or I can say adenosine monophosphate, guanylate, uridylate, and citylate. Now, it's not normal to have deoxyuridylate, and it's not normal to have thymidylate. Okay, now, so I said before that these nucleotides are linked to each other via a phosphodiester bond. So here you have a nucleotide right there, and the base is adenine. So this is an adenylate molecule. Why is it that it's not a deoxyadenylate? Well, again, you look at the sugar, it has a hydroxyl group, so you know that it is an adenylate molecule. Why is it adenylate, not adenosine? Because it has a phosphate group. Okay? Now, this is the phosphodiester bond that links this nucleotide to the next nucleotide. This is the third one. This is the fourth one. So that's a polymer. Now, does this polymer exist in DNA or RNA? It exists in RNA. Okay, why? Because the sugar is actually a ribose, not a deoxyribose. So what's the identity of these bases? Well, of the nucleotides. Well, you look at the bases on the side. Okay, so that's adenine, cytosine, guanine, and uracil. So we're only talking about the base itself. Okay, now look at the other polymer right here. Is it DNA or RNA? It's DNA because there is no hydroxyl group at carbon number 2, right? So this is DNA. And also you have a phosphodiester bond between the different nucleotides. The identity of these bases are thymine, guanine, cytosine, and adenine. Okay. So if I ask you what is the sequence of the nucleic acid on the left, you say it's adenine, cytosine, guanine, uracil. Or you can say it's A, C, G, U. So you can use the abbreviations, okay? Now, the other thing is that you never start from the other end. You never start, you do not say it's uracil, guanine, cytosine, and adenine. You don't say that. You always start, and this is the important point, you always start from the 5' end that exists right here. Okay, so let's zoom in right here. You start with the 5' end and you end up reading the sequence, the order of nucleotides to the 3' end. Okay, so there is directionality. Now, what's important before is that on one strand there is pairing between the bases. So whenever you have an A, on the other side paired with it you have a T. Whenever you have a C, you have a G. Okay, so these bases pair with each other very specifically. And there's a scientist known as Chargaff. He came up with a number of rules saying that in a DNA molecule, the number of A's is always equal to the number of T's, the number of G's is always equal to the number of C's, the number of purines is equal to the number of pyrimidines, because here you have a purine, here you have a pyrimidine. So whenever you have a purine, you have a pyrimidine. Whenever you have an A, you have a T. Whenever you have a G, you have a C, and so on. But the numbers of A's and G's may not be equal to the number of T's and C's. So basically, you can have a DNA molecule that is made of 99 A's on one strand. Of course, the other strand would contain 99 G's and 1 T, 1 C. Okay, so these are. So now the other thing is that the base pairing between the bases is different as well. So the base pairing between the adenines and the thymines is via 2 hydrogen bonds. Okay, whereas with guanines and cytosines, it's 3 hydrogen bonds. And this has very important implications when it comes to the stability of the DNA molecule. Now, you don't have really to know how they are paired chemically, okay? Just know that 2 for A and T, 3 for G and C. Now, so we say that the two strands are complementary to each other. And this is an important word for you to know. So DNA, the two strands of DNA, are complementary to each other, because whenever you have a C, you have a G. Whenever you have an A, you have a T. So we said that they are complementary, become لبعض. Okay. Now, the other thing is that in a DNA molecule, you have what is known as a backbone and side chains. The backbone exists of the sequence of phosphate followed by sugar, followed by phosphate, followed by sugar. So this is the backbone. The side chains are the bases because they go sideways. Okay, they go to the side. So these are the bases, or the side chains. Okay. The other thing that is important is that the two strands are linked to each other via hydrogen bonds, but they are anti-parallel to each other. What we mean is that if you look at the DNA molecule, if one strand on top you have the 5' end and the other end is 3' end. You look at the complementary strand, actually on top you would have the 3' end, and at the end you have a 5' end. Okay, so they are complementary to each other. They are anti-parallel, I'm sorry, to each other. Now, so if I say what is the sequence of DNA, you can say, well, it's 5' A, T, G, G, C, C, T, or you can simply just say it's A, T, G, G, C, C, T, etc. You don't have to say 5' when you tell me what the sequence of a DNA is, unless you want to start with the 3' end. So if you say A, T, G, G, I know that A would exist at the 5' end, and the other A right here would exist at the 3' end. But if you want to say 3' T, A, C, C, I know you're starting with the 3' end. You have to say it loud and clear that you're starting at the 3' end. Now, this is the sequence of RNA. It's A, U, G, G, C, C, U. So I know you're starting at the 5' end. Now notice something. With RNA, you have U. You don't have a T. In DNA, it's a T, not a U. Okay? And these two strands in DNA, this one right here and that one here, are complementary to each other, because whenever you have an A, you have a T. Whenever you have a T on one strand, you have an A on the other strand. If you have a G, you have a C, and so on. Okay? And notice that they are anti-parallel to each other. So this is 5' A, T, G, G, and the other one starts with 3', or it goes from 3' to 5'. If I ask you, what is the sequence of the complementary strand of the upper strand right here? You tell me it's TGAA. Again, if you say TGAA, I know you're starting at the 5' end, unless you specify 3' TACC. Okay, I hope this is clear. Now, DNA is flexible yet stable. So it's like an electric wire. This electric wire, you can bend it easily, right? But you really cannot stretch it and break it up. So the covalent bonds, the phosphodiester bonds between the nucleotides are really stable and strong. Okay, but otherwise DNA is really stable, but it can be bent. This is an important feature of DNA in terms of how it interacts with proteins, how it wraps around histones, and how it is packed in our in nuclei. Now, something about DNA as well, I said it's a helical molecule, okay? So it goes around like a helix, except that it is really not a perfect helical molecule. So it's not a smooth helical molecule. So notice the bend that exists right here. The reason why there is a bend is because of how these bases are stacked on top of each other. There is a degree of repulsion between a base and the base that comes right after it, right below it. Because of this repulsion, the base at the bottom would be tilted sideways and a bit away from the base on top. And that's why it's a helical molecule. Because it is a helical molecule, but not a perfectly helical molecule, you have what is known as these angles right here, and these are known as grooves. So right here you have a major groove, and right there you have a minor groove. Okay, زي الحفرة. So the order, if you look at one side of a DNA molecule, you have a major groove, major groove, followed by a minor groove, and so on. Opposite to a major groove, you have a minor groove. Opposite to a minor groove, you have a major groove. Now, this pattern is really important because this is how preferentially proteins interact with DNA. So you have this protein molecule right here. You can see that part of the protein fits into the major groove, whereas the minor groove is really too small for the DNA to put itself inside it. Okay, same thing right here in the major groove. So this is how DNA can interact with DNA. Now, this is like a zoom into how interaction really takes place. Now, one thing that I should have mentioned is that, so we can look here as well. So we can, as I said, the bases are exposed, even though they are oriented inward, some groups of the bases are exposed for the amino acids of the protein to interact with them. Another difference, actually, another difference is that in eukaryotic cells, chromosomes are enclosed into a nucleus versus a prokaryotic genome or chromosome that is not only it's circular, but it's not enclosed by a membrane, and that's why they are known as prokaryotic versus eukaryotic cells. Now, so in eukaryotic cells, DNA is really large, and it must be packaged into a nucleus. Now the thing is, so it's almost like in a single cell, in a single human cell, the length of DNA is about 6 meters long, and it has to fit into a small nucleus. So as an analogy, it's almost like fitting a 35-kilometer thread or little rope, thin rope, a 35-kilometer rope into a yellow tennis ball. So this is really humongous effort. So this whole DNA must fit into the nucleus, and in order for it to fit into the nucleus, it is wrapped around proteins known as histones. So whenever you have DNA, a single unit of DNA, and whenever you have it wrapped around proteins like histones, this is known as a chromatin molecule. So if I say a chromatin molecule, you know that I'm talking about a DNA molecule that is wrapped around or that is associated with proteins. In case of eukaryotic cells, these proteins are histones. And the basic unit of a chromatin is known as a nucleosome. Okay, so let's go into the nucleosomes and how they look like. Well, if you look at them, actually, you can see that you have a DNA that is wrapped around proteins, okay? And these are the histones. And then you have a DNA that is free of histones, and then you have another cluster of histone proteins and DNA wrapped around it, and so on. And then you have another cluster, and so on. Well, if you look at these proteins, if you look at this unit right here, just this one right there, it's actually composed of this DNA plus an octamer of proteins. Octa means 8. Okay, so basically you have doublets of 4 proteins, and these proteins are known as H2A, H2B, H3, and H4. They make up this octamer, and this octamer then allows for DNA to be wrapped around it. Okay, now there is another histone. Now someone might say, well, wait a second, I have H2 and H3 and H4, how about H1? H1, actually, histone 1 exists outside of the core particle right here. So this is known as the nucleosome core particle. Histone H1 exists outside of the nucleosome core particle. So it's almost like, that's how it looks like. It's almost like it seals the DNA. It fixes, it stabilizes the interaction between DNA and the nucleosome core particle. Now this whole thing right here, that is the octamer, histone H1, and the DNA that is wrapped around the octamer, this is known as a chromatosome. When you add the free DNA, which is known as linker DNA, now this is known as a nucleosome. Okay, so this whole thing right here, okay, so let's do this. Let's do this. So, okay. So this part, well, is known as a nucleosome. The octamer, the histone H1, the DNA wrapped around it, as well as the free DNA, is known as a nucleosome. Okay? Now, if you remove the linker DNA, if you remove this right here, and you keep only the octamer, the DNA wrapped around it, and histone H1, now this is known as chromatosome. Okay. Now, someone might say, well, wait a second. So what's the purpose of histone H1? What does it do exactly? It seems that it stabilizes the interaction between the octamer with the DNA. It does have another important function, and that is, if we look at human DNA or human chromosome, you would notice that some parts of the DNA are loose. That is, you have the DNA exposed, and some parts of the DNA you can have the DNA actually, there's no free DNA, there's no exposed DNA. Now the DNA that is loose is known as a euchromatin right here. Okay, this is known as a euchromatin. The DNA that is highly packed is known as a heterochromatin. Okay, so the difference between the two structures is actually determined, it seems to be determined by histone H1. So what histone H1 does is that it packs the DNA very tightly, or it forms the heterochromatin, in other words. Okay, so it seems that it really plays an important role in this. Now, this is important because when we later on talk about gene expression and transcription, pay attention to that. Euchromatins and heterochromatins. Okay, so someone might say, I'm a bit confused. So what's the difference between DNA, chromosome, and gene? Because I heard about what a gene and what is a gene exactly. So a DNA is basically the double helical molecule that is made of anti-parallel strands, two strands that are helically associated with each other via hydrogen bonds between the bases, between the complementary bases. So that's what a DNA is. Now, when you have a single large unit of DNA that is in eukaryotic cells, for example, that is wrapped around each other in histones. I'll show you when you have a whole unit of DNA, a large unit of DNA. Now this is known as a chromosome. Now zome, by the way, means object or body. Chromo means color. The reason why they gave it this name is that when they first identified it, they colored this molecule. So they said, this is the colored body. This is the colored object. So they called it chromosome. Now, chromatin is really part of the chromosome when it's part DNA, part protein. Now, a gene is basically an area within the DNA molecule or within the chromosome, same thing, within the DNA molecule whereby it can be transcribed. Okay, so there's a starting point and an end point, and we will talk about this in transcription. So if you have a long stretch of DNA, you can have multiple genes associated or found within these codes or within the DNA. Okay, so you have gene 1, gene 2, gene 3, gene 4, and so on. Now, we as humans, all living organisms having DNA, they have each organism, like human beings, we all have the same DNA, the same number of chromosomes, almost the same sequence of nucleotides within our DNA. We have the same order of genes. We have the same location of genes on the same chromosomes. Okay? There are subtle, little differences between us, but we have the same order of genes. So the same number and location of genes on chromosome 1, same order and sequence of genes on chromosome 2, and so on. But there are little differences in the sequences, and we'll talk about these differences. Remember, we are diploid, we are not haploid. What I mean by haploid is if we have one copy of each chromosome. Okay, so we say that these are 3 non-homologous chromosomes because they are different from each other. We have chromosome 1, chromosome 2, chromosome 3. Now, if I say this cell is a diploid cell, I'm saying that I have 2 copies, I have a pair of every chromosome. So I have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, and so on. These 2 chromosomes, and we said that these 2 chromosomes are homologous chromosomes. Okay, because homo, same, because they are same chromosomes. Same thing here. These 2 are homologous chromosomes. Okay, now why is it that these cells are diploid? Because one of them comes from the mother and the other one comes from the father. Okay, now what are the cells that are haploid in our body? Think about it. It's the reproductive cells, sperm and eggs. So RNA, very briefly, RNA is a single-stranded molecule usually. Okay, it's really long. It's unbranched because it's not a helical molecule. Now you do have the bases also oriented sideways, and you have a backbone, very similar to a DNA strand. Okay, now within an RNA molecule, you can have hydrogen bonds formed between the bases if they are complementary to each other. Okay, so and same rules apply to this hydrogen bonding as in DNA. That is A with U, remember, not T, G with C. The ribose sugar is a ribose. It is not a deoxyribose. Remember, we have uracil, not cytosine. It's usually single-stranded, not double-stranded. Now, RNA does not really have a precise structure. Okay, so it's different between one RNA molecule versus the other. So it's different than DNA again. There are different types of RNA molecules in our cells. Like maybe 20 years ago, 30 years ago, we only talked about 3 types of RNA molecules: messenger RNA, rRNA, and tRNA molecules. But then, in fact, there are different other types of RNA molecules identified. Each one of them has its own function. We'll talk about these different ones. The ones that are starred are the ones that you should know about. Other non-coding RNA molecules, we have a type known as long non-coding RNAs. We will talk about this type of molecule later on as well in our lectures. Okay, now you have the small nuclear RNAs. They function in a variety of processes, including splicing of the pre-messenger RNA. So we'll talk about these different RNA molecules later on.